Okay, thank you for the invitation. So I will talk about how patient data can allow us to test and model the cause of disease. And this is work with my collaborators, especially the first two students, Yashar Zegami and Inkyu Zhang, who are extremely talented graduate students, and Bratislav Misik have done a lot of the work that I'm going to present. So 25 years ago, little was known about the causes of neurodegenerative diseases. Now, however, we know that they're caused by prions. This is what Stanley Prusner said 20 years ago, and we can still say the exact same thing today. People still do not believe this model. However, the evidence is accumulating that all of the neurodegenerative diseases are caused by the propagation of an agent along the connectome. And this is true for Parkinson's disease, Alzheimer's disease, Huntington's, ALS, frontotemporal dementia, and I believe for schizophrenia as well. In the case of Parkinson's disease, the propagating agent is alpha-synuclein, which has the ability to become misfolded, and in its misfolded state, it is a prion that can cause neurodegeneration. So when we think of information flow through the nervous system, we know about neurotransmitters that transmit information rapidly. However, now that we know that proteins can be exocytosed and then endocytosed by the postsynaptic neurons, it raises the possibility that these proteins are also transferring information from neuron to neuron. And this would be slow information whose content would have to do with synaptic stabilization and pruning. And so the idea is that these prions convey bad information. They cause neurodegeneration by causing aberrant synaptic pruning. So Brack suggested that Parkinson's disease was due to a stereotyped progression of pathology from the brainstem up into the subcortical areas and then up into the cortex in these stereotyped stages. And he predicted that it was caused by a toxic agent, and in fact even predicted that the toxic agent started in the gut, which we now know is almost certainly the case. So we decided to test this model using this exemplary data set, the Parkinson's Progression Markers Initiative, which is funded by Michael J. Fox Foundation and NIH. And we used 380 patients and age-matched controls with anatomical MRI and extensive clinical and biochemical data. We used deformation-based morphometry to generate individual atrophy maps. And then we used independent component analysis. So we placed all of the patients and controls atrophy maps in a single four-dimensional set. And we used ICA, in this case we used Melodic from FSL, to generate patterns of covarying atrophy. And we found that one atrophy pattern was significantly different in the patients compared to the controls. And it is this pattern which we call the Parkinson's disease component. And atrophy in this component was correlated with disease severity on a whole slew of measures. But then the next question is this atrophy map a connectome? Does it reflect connectivity? So what we did here, we measured functional and anatomical connectivity. So each blue dot represents an anatomical region, a region of interest in the brain. And here we're measuring distance from the substantia nigra versus atrophy in every brain region. And you can see that for both a functional and an anatomical connectome, we get a fairly strong correlation. In other words, the closer you are to the substantia nigra, the more atrophy you show. And this is de novo patients, recently diagnosed patients. And now you can do this same calculation for every region in the brain and measure how likely it is to be the propagator. And when we do this, we find that the substantia nigra comes out as the most likely propagator, whether you use a resting state or a diffusion-based connectome. So we were interested in testing this further, and this is a paper that Yasser Itouria brought to my attention, which uses agent-based models of infectious disease spread. And here they're testing the spread of an infection across the globe, and they're testing how it is spread through air travel. So in each city, the infection spreads through diffusion, but then it will move from city to city via airplane travel. And here you have two viruses, H1N1 and SARS, that are modeled. You can see that, so what you can do with this is measure the arrival time of the virus. If the infection starts in a city, when does it arrive at every other city in the world? And you can see that physical distance is pretty poor at predicting arrival time. However, if you, instead of looking at physical distance, you look at topographical distance based on air travel, you get a very strong correlation between arrival time and connectivity via air travel. So we did the same thing with the brain. Instead of cities, we have brain regions, and instead of people with a viral infection, we have actual proteins. And we use the same agent-based model to model the propagation of misfolded synuclein through the brain. So this is Ingchu who did this work with Bratislav. We're testing actually two models here. We're testing the pathogenic spread hypothesis, where misfolded alpha-synuclein is a prion that propagates through the connectome and destroys cells. And we're testing it against the other hypothesis, which is that the pattern of disease in Parkinson's has nothing to do with propagation and everything to do with the fact that certain cells are more vulnerable than others. So here we use this SIR agent-based model. SIR refers to the fact that a person or an agent can be susceptible, that is healthy, infected, in this case a misfolded protein, or recovered, in this case meaning metabolized and removed. So we model each synuclein protein in the brain. They start off normal and healthy, and then we inject a certain amount of misfolded synuclein in one area and watch it propagate through the connectome. We also modeled selective vulnerability, and in this case we took synuclein concentration to be the measure of selective vulnerability in a brain region. And we used two genes, the SNCA gene, which is synuclein, and GBA, which is related to clearance of synuclein. So the amount of synuclein in any brain region at any time is based on the gene expression of these two genes, one that increases and one that decreases synuclein levels. So those are the only things you need to model. So gene expression plus a connectome, and then you inject the misfolded protein in one area and you watch it propagate, just like an infection. And what we found is that we could very strongly predict the amount of atrophy in patients. So you can see in panel C, comparing the empirical atrophy to simulated atrophy based on this model. And you can see that the correlation in panel B is very strong between the model and the actual data. You can also measure, try to find the epicenter by measuring something called the spread threshold, which is how much abnormal synuclein do I need to inject in any area in order for the infection to turn into an epidemic. And what we see here is that the substantia nigra on the right has the lowest spread threshold, meaning it's the most likely propagator in the sense that it can have the smallest amount of misfolded synuclein to generate an epidemic or a neurodegenerative illness. Here we also show that functional connectivity improves the model fit, meaning supporting the idea that synuclein secretion is activity dependent. We did permutation tests of both connectivity and gene expression to demonstrate that our model fit the data better than by chance. So these are null models of both gene expression and connectivity. And this supports the idea that the pattern emerges from two things, the connectome and the gene expression, which is a measure of cell vulnerability. In other words, the pattern is determined both by connectivity and local cell vulnerability. We also did an arrival time analysis, just like in the Brockman study, and you can see that the arrival time after injection in the substantia nigra, the map of arrival time actually matches the BRCA stages of Parkinson's disease. So the other thing we were interested in looking at is eigenmodes based on the work of Ashish Raj. And the idea here is that the structure itself determines the pattern of its destruction. The modes of destruction are embedded in the physical properties of the structure. And so he suggested this and tested it for Alzheimer's disease. So we did the same thing for Parkinson's disease. And in this case, we actually used partial least squares, where we had brain atrophy on the one hand in the patients and behavioral and clinical information on the other hand, and looked for patterns of covariance between the two. And we identified a latent variable. And the first latent variable that we identify with this looks exactly like the atrophy map that we had identified previously using ICA. Now it's the same data set, so it's not necessarily too surprising. But with PLS, we identified this one mode of degeneration that on the right you can see is correlated to almost all of the clinical measures of disease severity. Interestingly, we find two other modes of disease. So first of all, the first latent variable, for the score for each individual, predicts disease progression over 5 years. So at diagnosis, the amount of atrophy in this first latent variable is predictive of disease severity. But as I said, we also identified a second and a third latent variable, and we think that these represent a benign mode and a severe mode of propagation. So on the left, you have a benign mode associated with mild symptoms and mild clinical signs and lack of dementia. And on the right, sorry, the slide didn't work out, but on the right you have a malignant mode. That is a mode of disease spread that would be severe and is associated with more severe symptoms, dementia, and postural instability, markers of disease severity. And they both have different patterns. So the benign mode appears to be associated with atrophy of motor areas, whereas the malignant mode is associated with limbic atrophy. And indeed, there is evidence that when the disease propagates to the limbic system is when you start to have dementia in Parkinson's disease. And then the final little thing I want to show is this test of the network spread hypothesis. So here we used a second MRI scan at 1 year, and the prediction is that the disease severity or the atrophy pattern at time 1 would predict the atrophy pattern at time 2 based on connectivity. So we start with time 1. We call this the disease reservoir with the analogy to infections. So these are the areas that are affected at diagnosis. And then the prediction is that cortical thinning from time 1 to time 2 would be proportional to disease exposure, which is based on connectivity and atrophy. So any voxel or any vertex in the cortex, its connection to the disease reservoir multiplied by the amount of atrophy in the disease reservoir would determine its likelihood of being thinned. In other words, its exposure to the infection. And indeed, this is what we found. We showed that whether you use a functional or an anatomical connectome, there's a strong correlation between disease exposure measure and cortical atrophy, supporting the disease spread hypothesis. So in conclusion, and I realize I've been using the word predict and I've been using it wrong, and I apologize to Gael, but in conclusion, I think this shows how we can use patient data, especially good, open source, well-characterized, and high-quality data, to generate models of disease that allow us to test specific theories and pathological mechanism. And in the case of Parkinson's disease, I think our work shows that this combination of connectivity and synuclein concentration is very strongly supportive of the prion hypothesis in Parkinson's disease. Thank you very much.